Morning. Okay, so Hodges is here. Okay, where are we at here? Yes. 0.5. All right. And again, let's see. Like I said, Hodges is physically here. So is Midkiff and Rice. Richardson is online. Taylor is here. Michaela is here. Asbury is online. Tudor is here. Burdette is online. And that's what we have so far. We have about 2 minutes left. Are there any questions? Since we're recording, I don't mind answering questions a little bit early. One question that was asked before we started recording was about next week's lab. Next week's lab will also be virtual. And I still plan to be here physically. And that is still the plan, is to physically come into lab next week. But of course, if that changes, you'll know about it. And as always, I always recommend getting the labs done early. Cedric is online. Eric is online. One thing I might start doing next week. Massey is here. Starting next week, if you're online, I might need you to, it's funny that should just happen. I might need you to have your camera on. Because some people who are consistently online, at least on paper, still seem really, really lost. And there's no way you're paying any attention and asking some of the questions you're asking. So I'm thinking, I'm just trying to figure out ways that you're cheating, getting around the system. Like maybe you're just like, look, you log on, go back to sleep. I'll stay up. I'll give you the words and the instructions, and you send them in before 9. That's got to be what's happening for some people. So I'm just trying to avoid that. And again, like I've said, if you come in, you get extra credit just for physically being here. I'm trying to incentivize you. Some people really, really see, you don't need to do it yet. It's like you guys read my mind, though. That's awesome. Anyway, Felder is online. So yeah, starting next week, that's probably what's going to start to happening, is you're going to need to have your camera on when you're online. Obviously, I don't expect you to be there the whole time. Sometimes you might need to pick your nose, go to the bathroom, whatever. So sometimes I'll understand if you're away from your computer. I'm not going to sit there and stare at you. Your thing's not going to be in front of the screen. As a matter of fact, no one else is going to be able to see it unless you're already online. So that might be what's happening. I don't know. If anyone else has some ideas of how I can ensure people are actually paying attention, let me know. Because again, some people are doing really, really poorly because they're just missing out on information that I've given 1.5 million times. Speaking of information I'm giving out 1.5 million times, please read the announcements. Some of you have like 5 independent work points. In the very least, if you read the announcements, you can see where it says, hey, if you read this, reply to the email and get independent work points. So read the announcements, at the very least, to get your independent work points. But again, I'm getting questions from people who are failing. And they're like, hey, what can I do this? Like, yes, I've been asking you to do this all semester. So please read the announcements. Please pay attention to lecture. I'm just trying to help you. And for the rest of you, I'm sorry you're dealing with this stuff because you're doing okay. And you're probably like, I know. You say this every day. Please shut up. So I apologize if you're doing good and you're hearing me preach. But I'm really trying to get those people who are failing to pull it up, especially before Friday, because Friday is the last day to withdraw. And I strongly recommend, if you have a D or below, try to find the time to meet with me, or just really look at yourself and be honest with yourself and really think. It's not that hard to pass this class, right? It's not that hard to maintain a C. Pay attention, do what you're told. But it's even harder now if you've got to go back and fix half of a semester's worth of mistakes. So you have a big road ahead of you. It's possible, but you've got to be ready to do it. So Friday is the last day to withdraw from classes. And again, that's a good point, right? Read my announcements. Make sure you pay attention to the lectures. Friday is the last day to withdraw from the course. So any questions about anything? All right, let's jump into it. So I was looking at, actually, let me fix this really quick. I was watching that video as I was making the questions for it for Monday, and I thought, you know what? We should finish the last part of this chapter. So I'm going back to chapter 9 so we can finish chapter 9. And then we'll jump into chapter 10. Oops. Then we'll jump into chapter 10. Oops. And do this really quickly. Entire screen. Okay. So, let's see. Got it. Okay. Slideshow. There we go. So we're going to talk about sex-linked genes. This won't take long, and then we'll jump into chapter 10, which is the last chapter of this third exam, right? The third exam is going to be chapters 8, 9, and 10. So once we're done with this chapter, we're going to take another exam. So go ahead and start thinking about what day you might want to take it. Obviously not next week. I know we've got somebody out of town, so we definitely won't do it next week. It'll probably take all next week just to finish chapter 10. So the first word for attendance will be humans. If you're watching the live stream, or maybe the video, we'll see. I'll probably have questions for the video, but just in case, take a screenshot of me holding up number 1, I guess you could say, and also pointing at the word. So whether you're watching this right now or watching the video, I need a screenshot of me doing that. Make sure that's the whole screenshot. So it shows your entire screen, not just zoomed in on me, right? That way I can see, okay, this person was on the phone, this person was on a computer with these tabs open. That way I can differentiate your screenshot from someone else's. Again, I'm just trying to make sure everyone's paying attention. So let's talk about sex-linked genes. Some of the stuff we already said. This part we already said, right? A sex-linked gene is simply a gene that you would find on one of the sex chromosomes, right? One of the non-autosomes. Most of them, as we already said, are on the X chromosome as opposed to the Y chromosome. I already told you why. Just look at the size. There's the X chromosome. There's the Y chromosome. Obviously, there's a lot more chromatin, right? There's a lot more DNA here than there is here. So it stands to reason there's more information on the X chromosome. Not that I'm going to ask you that. I'm definitely not going to ask you which one has more genes on it. But, that's a good independent work topic. Not that you need to know this, but you could write about it. How many genes are on the X chromosome? How many genes are on the Y chromosome? We're obviously about to talk about some of them. Some human conditions, including red-green color blindness, hemophilia, and some other ones that are not listed, and a type of muscular dystrophy are a result of a sex-linked recessive allele. So again, another independent work topic. Here I'm listing some sex-linked things. Look it up for the other ones. And if you watch that genetics video from the genetics lab, you know at least one of the other ones. So yeah, look into that if you want. So let's talk about red-green color blindness. Before I start talking about it, I'll go ahead and tell you I'm not specifically going to ask you any questions about red-green color blindness. I just wanted to talk about it so you understood how this works in general. And because, again, I teach out of the book. This is a common sex-linked disorder caused by a malfunction of light-sensitive cells in your eyes, and it mostly affects males. So hopefully, if we had all the time in the world, and I could turn off the screen and we could talk about it. It's like, why does it mostly affect males? Hopefully we could come to the conclusions I'm about to give you. Like I should ask, all right, do you think it's on the X chromosome or the Y chromosome? What do you guys think? Not that we have a lot of time to sit and think about it. But if it affects mostly males, do you think it's on the X chromosome or Y? That's okay. That's a thinker if you don't normally think about this, but I'll get to it. But here's a hint. Heterozygous females have defects too, some defects. So if you're a female and you're heterozygous for this condition, you're not necessarily colorblind, but you do have some issues. And again, that's another independent work topic if you want to look into it. What are the other issues? So the next slide I'm going to show you shows a test for red-green color blindness. Can anybody not see this number? Of course, if you're online, you might not be able to. It might be blurred a bit. So everybody here can see it? So none of you are colorblind, at least not red-green colorblind. That's a number 7. That's a typical test. So because they are located on sex chromosomes, sex-linked genes exhibit unusual inheritance patterns, and we're about to talk about why. And before I do talk about this, I want to back up and remind you of some of the things I've said earlier, and I've used specific wording on purpose. So when this chapter first started, I used to say to have the recessive phenotype, both of your alleles had to be recessive. And then I started to move away from that, and I said the same thing in a different way. Then I said, if you have the recessive phenotype, then all your alleles need to be recessive. Does that make sense? So in one sense, that's almost saying the same thing, right? Because if you're talking about chin dimple, if you don't have a chin dimple, that's the recessive phenotype, right? So you would have to have 2 recessive alleles. Make sense? But another way of saying you have 2 recessive alleles is saying all of your alleles are recessive. However, things are a little bit different with sex chromosomes, and this is why I say all your alleles now, and you'll see why. So here we go. Let's look at this genetic cross, this Punnett square. Here we have, let's see, a female, obviously, because it's an XX and a male, XY. This female has completely normal vision. It's not even a carrier for colorblindness, completely normal. This male is colorblind. Now notice, they only have 1 recessive allele. That's kind of what I was getting back to. I'm getting back to what I was saying earlier. Because in this case, it's not that you have 2 recessive alleles to have the recessive phenotype. The more accurate way of saying it in this case is all your alleles are recessive. That makes sense. Make sure that makes sense before we move forward. Because this person, because they only have 1 X chromosome, because it's a male, they're only going to have 1 allele. It's either going to be dominant or recessive. So when it's recessive, all their alleles are recessive because they only have 1. Hope that makes sense. So anyway, if you do the cross and you look at the possible outcomes, right, there's a 1 in 4 chance of having a female XX who's a heterozygote. Excuse me, there's a 2 and 50% chance of that happening. So having a heterozygote female and a 50% chance of having a normal male. And again, I'm not going to test you on this, so I'm not going to take any questions, and I'm not going to explain it any further. But hopefully by now you're very familiar with doing these Punnett squares, because you might have to do a couple on the exam. If you do, they won't be for anything sex-linked. Now here's a different situation. So the last situation again was a normal female and a colorblind male, and there was 0% chance that any of their kids would be affected. Here we have a carrier female, so she's technically normal, but she's a carrier. She has 1 of the recessive alleles. Here we have a normal male, right? So unlike last time, we had a colorblind male. This time we have a normal male. Now look what happens when they mate. You do the Punnett square. There's a 25% chance they'll have a completely normal female, a 25% chance you'll have a completely normal male, a 25% you have a heterozygote female, and a 25% chance that you'll have a colorblind male. So again, you have a 1 in 4 chance for each one of them, right? There's an equal opportunity for any combination. And again, I'm not going to ask you this, but it's a great way, a good review to look at Punnett squares. So any questions about this slide? To me, an interesting thing about this is, if you didn't sit down and do the Punnett square and you had to guess, and I said, all right, again, think about this last one. We have 2 couples. The first couple is a colorblind man and a normal female. The other couple is a carrier female and a non-colorblind male. Which one's more likely to have a colorblind kid? And to me, if you didn't think about it, I would expect this couple to have a colorblind kid, right? Because you're like, oh yeah, the father has colorblindness. And then this one, there is no parent who's colorblind. But yeah, when you look at it, it turns out that there's more of a chance in this situation. And basically, as far as I'm concerned, what this should tell you at a 100 level non-biology major class is that sex-linked genes are weird, right? They're not like you would normally expect. But if you were to take the time and do the Punnett squares, they're still just as easy to figure out. And of course, here's a situation where we have a carrier female and a colorblind male. Now you have a 50% chance of having a colorblind kid, and a 25% chance of having a carrier female, and a 25% chance of having a normal male. Not that I'm going to ask you any of this on the exam. So are there any questions about red, green color blindness? All right, the next thing we're going to talk about is hemophilia. Put a big old X to this too, because I'm not going to ask you about this on the exam. But it's a great example because it also involves history. So if you're a history buff, unless you're very specific about what kind of history you like, then this might be interesting to you. Because hemophilia has changed the course of human history. So what is it? Hemophilia is a sex-linked recessive trait that has a long, well-documented history. What are hemophiliacs? They are people who bleed excessively when injured. They have abnormal allele for blood clotting factor. And the most seriously affected may bleed to death after relatively minor bruises or cuts. Do any of you know anybody who has hemophilia? Yeah. So it can be dangerous, right? I actually know somebody too. Because if you get a little bit of injury, what might be okay to us could kill somebody else. Of course, medical technology being what it is today, it's not as dangerous as it used to be, but it's still a little bit dangerous. And again, it used to be very, very dangerous. So again, like I said, it has a well-documented history. Why is that? Well, because there's the age-old practice of strengthening international alliances by marriage, right? So if you know anything about pre-World War I Europe, for example, it was all about kissing cousins, so to speak, right? So to make sure everything was copacetic between the countries, this person mated with this person and this person married that person. And a lot of the times there was a lot of inbreeding going on. So first of all, it was well documented because they were royal families. And second of all, even though it may have been rare in the normal population, it wasn't rare here because they were all mating with each other. So yeah, you can look into that if you want for independent work. I'm going to scratch the surface, but look into it a little bit more. Like, how did hemophilia affect world history? So here's an example. I'm definitely not going to ask you questions about this on the exam, like I said, but here we're looking at the royal family of Russia prior to the revolution. So you can see Queen Victoria was a carrier and married and had kids with regular old Albert. And then they had a carrier female who had kids with a regular old Louis. And they had another carrier female who again married a regular old guy as far as hemophilia is concerned. And then finally they had a son who was affected. Right. And the reason, and not that this matters for biology, but in case you're curious, one of the reasons this is so important is because Alexis, the kid here with hemophilia, it was dangerous. So they had this guy, some wacky guy there for purposes of treating his hemophilia, but he was really crazy. And a lot of people thought that that this guy had the royal family's ear. So they're really suspicious of him. So eventually they overthrew the royal family, killed a lot of people, including the kids. And that was the Russian Revolution that ushered in communism, which obviously has changed the world since then. So look into it if you want. Not just that, but what are some other times biology has changed history? Think about, too, the fact like when we came over to America, right? So when Europeans came to the New World, they also brought their smallpox with them. Think about how many Native Americans they killed and how, if that didn't happen, how we may not have been able to take things. I say we because I'm sorry to assume, but you all look Caucasian to me. And I'm half Caucasian, I guess. Anyway, yeah, so look into that if you want. And that's in chapter 9. Any questions about chapter 9? All right, let's quickly jump into chapter 10. And 10 will be the next attendance word, the number 10. So if you're online, send me a picture of 10, right? 10 of something, or maybe you can find the number 10 drawn or typed somewhere, but don't actually type me the number 10. I want a picture of the number 10. There's also a Pearl Jam album called 10 that would count. So again, that is the second attendance word. It is number 10. Here we go. Let's start chapter 10. This is the stuff that would have been really relevant for this week's lab, because this week's lab is all about DNA. But lucky for you, this week's lab is really, really easy. The hardest part of this week's lab, if you haven't done it yet, is when you first click that link and it takes you to the website, is to actually find the other link that takes you to the simulation. That's it. And then after that, you just follow the directions and answer the questions as you go along. It's so easy. Anyway, But here we go. We're talking about the structure and function of DNA. Another thing I want to remind you is we've already talked about this. In chapter 3, we talked about biological molecules, and one of the whole sections of chapter 3 was nucleic acids. So a lot of this is going to be review, which should help. All right, why molecular biology matters. Just to save time, let's go through it. I won't even ask you, because I don't even remember some of these. This is an example of sickle cell. So we talked about sickle cell in the last chapter in the context of, actually, I don't even remember what the context was. But yeah, we talked about sickle cell in the last chapter. We're talking about it again in this chapter. Then we're talking about it again in the next chapter. But basically, sickle cell is caused by one single mutation in the genome. And what you're going to learn about is what a genome is and what a mutation is. And that's why it's relevant to this chapter. I assume, and I don't remember for sure, this person's maybe making some insulin or making some vaccines, but to be able to do either one of those things, you have to understand DNA technology. And then I don't even remember what the cow situation is. So yeah, let's just skip through this. This is always just a fun little game. Nothing's important on this slide. Let's jump into chapter 10. Actually, let me say this too, if we're talking about why molecular biology matters. As far as today's situation is concerned, this one might be the most relevant chapter that we do. Because we're still sort of in the COVID-19 pandemic, right? Yeah, we're kind of easing out of it and you don't hear much about it, but it's still around. And hopefully it's still fresh in your brain. So to truly understand any kind of virus, you need to understand chapter 10. And to truly understand most vaccines, but especially the mRNA vaccines, you need to understand this chapter. But this is very relevant, probably the most relevant chapter to your everyday life. You're not going to be an expert on any of this, though. This is just scratching the surface. So, when you're done with this chapter, don't feel like you're going to be able to debate COVID-19 vaccines with anybody. It's not. This is just the foundations. So when you do, if you get interested in looking into this stuff, you'll have a good foundation to start. You won't be starting from scratch. Your book always starts with this, Biology and Society, and they talk about something relatively recent that is relevant to this chapter. This book was published before COVID-19. Otherwise, I'm sure this section will be COVID-19. And I'm sure the next version of your book, this will be about COVID-19. But at the time, Zika was the most recent of the new viruses. So they talked about Zika. Again, I'm not going to ask any questions about it. You can look into it if you want. Long story short, your book is saying what I just said, which is that if you're going to understand viruses and certain pandemics, obviously, that are caused by viruses, then you're going to need to understand what's in chapter 10. And that's why this is relevant to your everyday life. So here we go. This chapter is broken down into 3 main bullet points. First of all, we're going to talk about DNA structure and replication. And again, a lot of that will be review. You definitely know the structure to an extent because we talked about that in chapter 3. But we're going to talk about it again and get a little bit deeper. And for replication, thanks to chapter 8, when we talked about mitosis and meiosis, you know that at least you already know that replication happens in the S phase of interphase. So you already know that. But then we'll dig a little bit deeper there too. Then after that, we're going to talk about going from DNA to RNA to protein. And then finally after that, we're going to talk about once you know all that information, that background information, then we can finally talk about viruses and other non-cellular infectious agents. So here we go. Your book kind of teaches this in a history form. I like how they do this, sort of like they did with Mendel, right? Instead of just telling you how Mendelian genetics work, they kind of did it like a story. Like, all right, Mendel, he did this at this time, and then he did this at that time, and then he learned this, and then he learned that. And we're going to do kind of the same thing. But for the exam, I'm not going to test you on the history of what we knew, right? So basically what I'm saying is so far there's not really much to write down yet. This is just like an introduction. Like, let's teach you how we figure this out as a human race before we get to what we know now. So yeah, if you're taking notes, you give your note-taking hand a break. But yeah, where did we start? In the 1800s, we knew the DNA was in cells. That was it. Like, so forget what you know about DNA right now. Back then, all they knew was there was something called DNA. Of course, I don't remember if they called it that. No, they didn't call it that yet. But they knew that there was this stuff, and they knew it was in the cells by the late 1800s. Then by the 1930s, somebody had figured out that there was a certain molecule that was responsible for inheritance, right? So Mendel had already come at that point. Mendel had already figured out this inheritance that you just learned in the last chapter. But then in the 1930s, someone figured out, all right, there's a molecule, some kind of molecule responsible for all this inheritance, right? All the stuff we learned about in chapter 9, right? Someone finally figured out in the 30s, all right, there's a certain molecule responsible for that. Then in the 1940s, people figured out more about chromosomes, right? So they knew chromosomes were in the cells. But in the 40s, they finally figured out, all right, we know what chromosomes are made of now. They're made up of about half DNA and half protein, like I've already told you, right? So the DNA wraps around histones, and each one of those little balls is called a nucleosome. That's a review for you. We've already told you that. So again, that's what they figured out in the 40s. So before I move to the next slide, look at everything we knew by the 40s. None of those dots were connected yet, right? We knew there was a molecule that was responsible for inheritance. We knew a little bit more about what DNA was and what chromosomes were. But at that point, no one knew that DNA was responsible for inheritance yet. Then in the 50s, they finally figured out that it was DNA that was the molecule responsible for heredity. So in the 1950s, they put all those things together and said, okay, Mendel figured out all this inheritance stuff, all this genetics. And then in the 50s, they said, and it's this DNA that we've been studying. That's the stuff that's responsible. And once they figured that out, that ushered in what we call molecular biology. We'll put a question mark next to this, which means I'm probably not going to, go back. I'm probably not going to ask you the definition of molecular biology, but I might. Most likely, it's a really slim chance that I will, because this whole chapter is basically, they could be titled molecular biology, because that's what you're learning. It's the study of heredity at the molecular level. So the last chapter was the study of heredity, right? But then we were just looking at patterns, like, all right, if this thing mates with this thing, then we could expect these offspring, right? We were looking at the overall picture. That's the kind of studies you could do without a microscope, right? It's like this thing and this thing mate, look at the offspring, take the data, look at the numbers, right? You don't need a microscope to look at that. Now we're zooming in, right? So now we're going to zoom in and look at this inheritance from the molecular level, hence the name molecular biology. Any questions so far? Okay, so that's a good introduction. So again, everything I just told you is just kind of a segue into what I'm going to teach you. None of that's necessarily going to be on the exam. So once they figured out that it was DNA that's responsible for these inheritance patterns, the next effort was to figure out the DNA structure. Like what I said, starting back in chapter 1, that's going to be a theme of this whole semester, right? Structure and function, structure and function. To really figure out how DNA functions, how is it responsible for heredity? Figure out its function, and they had to figure out its structures. That's where they started, was trying to figure out its structure. At this point of the story, people knew all the atoms and how they were bonded. So they knew, like, all right, this carbon is single bonded to that carbon, which is double bonded to this oxygen, right? They knew all those things. But what they didn't know was the three-dimensional arrangement. Again, this is still a segue. Sorry, I should have said that before we started. Nothing here is going to be on the exam either. This is still a segue leading us to the stuff you need to know. So again, at this point in the story, they know DNA is responsible for the heredity. They know all the parts. They just don't know the three-dimensional arrangement of it. But that's what they need to figure out, because again, structure, function, right? Because what they wanted to know was how. How does it store this genetic information? How does it copy the genetic information and then pass it from one generation to the next, right? They didn't know all this. They knew it did it. They knew that was the molecule responsible for it. They just didn't know how it worked. So that's why they tried to figure out the structure. Excuse me. Anyway, these are all great independent work topics if you want to look into it. So, exactly at what point did they know what all the atoms were and how they were bonded to each other, and who figured that out, and how did they figure it out? And it can be said for all the other stuff I told you. Who figured it out and how did they figure it out? So any questions so far? Again, as far as the exam is concerned, this is all unimportant stuff. It's just a segue into the important stuff. So here we go. Speaking of the important stuff, let's jump into it. Let's talk about DNA and RNA structure. As you already know from chapter 3, and as you need to know for the exam, DNA and RNA both are considered nucleic acids, right? So they're not carbohydrates, they're not proteins, they're not lipids, they are nucleic acids. That is the category of biological molecules that they are, nucleic acids. Of course, just to step back and remind you, chromosomes themselves are DNA wrapped around protein. But we're not talking about chromosomes yet. Just talking about that DNA molecule. So yes, they are nucleic acids. And as you know from chapter 3, and as you're going to need to know again, you need to know that relationship between monomer and polymer, right? Monomers make up polymers. The monomer in this case, whether you're talking about DNA or RNA, they're called nucleotides. And the polymer is called, luckily enough, it's very easy, polynucleotide. So DNA and RNA, the long part, the polynucleotide, they're all made up of individual nucleotides. And if we're talking about DNA, then they can have any sequence of 4 types of nucleotides, which are these 4 right here, A, T, C, or G. Of course, the same thing can be said for RNA. They can also be very long and have any sequence of 4 types of nucleotides, except in this case, instead of having a T, it would be a U. And don't worry, this will be on the screen again and it'll look better. But yes, nucleotides are the monomers, polynucleotide is the polymer. There's 4 different types of nucleotide, regarding of whether you talk about RNA or DNA. If you talk about DNA, the 4 types of nucleotides are A, C, T, and G. If you talk about RNA, the four different types are A, C, U, and G. Now I'm going to move forward. So don't worry. If you have questions about that, it's going to come up again. So I'll show you again. Are there any questions about this slide? All right. So let's look at the nucleotides. Here we go. Here's the DNA double helix. You can see this is one strand, right? That's one polynucleotide of DNA. This is a separate polynucleotide of DNA, right? Those are 2 strands of DNA that are together. Remember hydrogen bonded, which we talked about in chapter 3. We'll talk about again later. And then twisted together, right? So what you're looking at here is 2 strands of DNA. So if we untwist it, we can see what we're talking about. Well, we didn't necessarily untwist it here. We're just looking at one strand in this picture. This is one way of looking at it. There's an A, there's a C, there's a T, there's a G, there's another G. So again, you can see that this is nucleotides attached to each other to make a polynucleotide. This picture right here on the very right, it's the same thing as the one in the middle. It's just drawn a different way, right? So this one's more about showing you the shape of the molecules. So they're showing the same thing, just showing it different ways. There's nothing to study on this picture. It's just an example. This is just showing you the strands of the polynucleotides of DNA. Any questions about that slide? Okay. Let's talk, like I said earlier, let's talk about these nucleotides. As you already need to know, these nucleotides are joined by covalent bonds. Specifically, and I said this in chapter 3, and I said in chapter 3 you didn't need to know it, but you would need to do it later, so I'm telling you now. You did need to know it was covalent bonds. But now I'm being very specific. And you need to know that it's the sugar of one nucleotide that's connected to the phosphate of the other nucleotide. Because remember, and I'll bring this back up later too, a nucleotide is made up of 3 parts, like we talked about in chapter 3. There's the sugar, the phosphate, and the base. So it's the sugar and the phosphate that are bonded to each other. The base is just hanging out there. And you can see it in this picture, right? Sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. The bases are just out there kind of doing their own thing. At least that's what we'll say for now. They're doing something, but we're not going to talk about what yet. So again, you need to know that the nucleotides are covalently bonded with each other, from the sugar of one to the phosphate of the other. Hence the name sugar-phosphate backbone. What I won't do is give you this picture on the right and say, all right, which one's the sugar and which one's the phosphate? Right? So yes, that blue one's the sugar and the yellow one's the phosphate, but I'm not going to ask you to differentiate. Speaking of which, the next attendance word, you're writing it down, like for you guys, the person who just want the extra credit, it's 2 words, blue, yellow. But if you're online, send me a picture of something blue or something yellow. Doesn't matter what it is, I don't care. Just send a picture of something blue or something yellow. Anyway, any questions about this slide? Like I already just said in the last slide, and like we already said in chapter 3, the 3 parts, the makeup of a nucleotide, are the nitrogenous base, the sugar, and the phosphate group. And you need to know that. We're about to talk about each one of them in a little bit more detail. But if you're taking shorthand, it would be okay to just memorize it as, whoops, forget that, sorry. Base, sugar, and phosphate. Yes, technically it's a nitrogenous base, and yes, technically it's a phosphate group. But for taking notes and memorizing, like if you're doing flashcards, you'd be fine memorizing base, sugar, phosphate. And there's a picture of it, right? Here's the sugar, here's the base, and here's the phosphate. And we're going to talk about all 3 parts. So again, before we dive into the 3 parts that make up a nucleotide, I want to remind you again of the big picture. This is one nucleotide, right? And the one nucleotide, these things, you string them together to make a polynucleotide. So let's talk about this stuff right here. The nitrogenous base. Let me just circle the only part that you need to know before I get into it. The rest of it you don't need to know, but we'll talk about it very quickly. The nitrogenous base projects from the backbone, because remember, like I said, it's the sugar and the phosphate are covalently bonded together. So that's the backbone. That's the thing that holds the polynucleotide together. And the nitrogenous base just kind of hangs off the side of it. Technically, it has a ring of nitrogen and carbon, as you can see right here. But again, I'm not going to ask you that. It also has various chemical groups attached to it. And it's that arrangement, right? It's the arrangement of the carbons and nitrogens and the various chemical groups that make it, an A versus a C versus a T versus a G. And I'm not going to ask you that. And actually, it's not on the slide either. But you could define, if you will, you could define a nucleotide by its base. Because the sugar and the phosphate, like if we're talking about DNA, A, T, C, or G, doesn't matter. They all have the same phosphate group and they all have the same sugar. The only part that's different is the base. And the base is different because of the shape of it and the various chemical groups attached. And of course, as your book points out here, it is also basic, right? It's not acidic, it's basic, hence the name nitrogenous base. But for the exam, again, all you need to know is there's 4 different versions of nitrogenous bases, A, C, T, and G. And we're going to come back to those. So any questions so far about nitrogenous base? All right, the next part is the sugar. And really, the only thing you need about the sugar is that it's a sugar. That's it. That's just one of the 3 parts. Everything I'm about to tell you, I'm not going to ask you about on the exam. It's good to know it, but even if you forgot it, you could completely understand the rest of the stuff in this chapter and the rest of the stuff in the semester. So in my book, that makes it unimportant. Remember, sugar and phosphate are bonded together. The rest of the stuff that I'm about to teach you, who cares? But anyway, it's in the book, so here it is. The sugar has 5 carbon atoms, 4 of which are in the ring. One of them extends above the ring. And then that's right there. That's a bad picture in that context. And the ring also includes, yes, one of them extends above the ring. And the ring includes oxygen. Hence the name. Well, never mind. We'll just leave that alone. So again, that's the sugar. I'm not going to ask you any questions about all the stuff that's right here that I put it next to. It's good to know if you want. But again, this information is not important for you to understand everything I'm about to teach you. The other stuff is. So any questions about the sugar? All right, the next part is the phosphate group. And again, just like the sugar, you just need to know that there is a phosphate group. And of course, like I said, it's the sugar and the phosphate. They covalently bond to each other to make the sugar-phosphate backbone. But again, here's the information from your book, so I'm going to teach it. You don't need to know it. But the phosphate group has a phosphorus at the center, go figure. It is the source of the acid in nucleic acid because unlike the nitrogenous base, it is acidic. And it has a negative charge on one of the oxygen atoms. So again, all of this information would be relevant if this was not a 100 level biology course. And this was not a biology course for non-biology majors, because that stuff does come into play later, much later in your educational career if you're studying biology. But for now, you don't need to know that. Everything I'm about to teach you, you can understand without knowing this stuff right here. Are there any questions about the three parts of the nucleotides? Okay. Like I said earlier, there's 4 different types of nucleotides, specifically 4 different types of nitrogenous bases, right? So again, nitrogenous bases are made up of 3 parts. The only part that's unique to each one is the nitrogenous base. The sugars, the phosphates, they're all the same. So what are the 4 different nitrogenous bases? Here they are again, T, C, A, and G. Specifically, thymine, cytosine, adenine, guanine. Now your book points out that some of them are single ring and some of them are double ring. You do not need to memorize that. You certainly don't need to memorize which one are singles and which one are double. Just lock that in the back of your brain. Just the fact that some are single and some are double. That itself will come up later, but you don't need to memorize which is which. And when it does come up later, it's really not going to be that important. It's just going to be a little important. So yes, even though I presented it in this way, the T and C are single rings and A and G are double rings. Even though I presented it in that way, you don't need to memorize that. It'll come up later, but it won't be that important. What's important on this side, as far as the exam is concerned, is that there's 4 different types of nucleotides for DNA. They are T, C, A, and G. Another thing I'm not going to ask you is to, I won't say, there won't be a question where it says, all right, what is a T? And I give you 4 different choices of words that start with T, right? I'm not going to do that. You don't need to memorize that the T stands for thymine or the C stands for cytosine. I'm not going to do that. It's a 100 level non-biology major course. So any questions about the 4 different types of nucleotides? Okay. As you already know from chapter 3, RNA and DNA have some similarities, but they also have differences. Just like in chapter 3, you're going to need to know them again for this chapter. So for this next exam. Also, this should be a big hint. If it's on the first exam and it's on the third exam, there's a higher probability it's also going to be on the cumulative final. Anyway, let's look at some of these differences. Like we already said, DNA has A's, C's, T's, and G's. RNA does not, because RNA does not have T's. They have U's. So there's any question on this exam, and there will be, where it says anything about RNA, like which one of these is the proper RNA molecule. If it has a T in it, you can just go ahead and eliminate that and don't even waste your time figuring it out. You'll see what I mean later. But if it has a T in it, it's DNA. If there's any questions asking you for RNA, if it has a T in it, the answer is that's not the answer. So you can save some time on the exam. When the question asks you for RNA, you can just completely forget, cross off the options that have T. Another thing, it's not really important for a 100 level non-biology major course, but I'm going to go ahead and tell you anyway. RNA, like I said in chapter 3, has a different sugar. The sugar is called ribose as opposed to deoxyribose. And the only reason that would be relevant for you guys is because that's just explaining how they get their different names. RNA and DNA, right? Ribonucleic acid versus deoxyribonucleic acid. So they have different sugars. RNA has one different base, and they both have different sugars. Any questions about that? To be honest, there's not going to be many questions about this either on the exam. You'll see. We haven't even gotten into the good stuff as far as the exam is concerned yet. A lot of the stuff for the exam is going to be that middle bullet point, that process of going from DNA to RNA to protein. That's going to be the meat and potatoes. But here we go. Let's move forward. If you download the PowerPoint, you can watch this little animation, kind of highlights the differences between DNA and RNA. I'm going to share some more videos on Google Classroom that you can watch for independent work. Those are better than this. This is just what happens to come with your textbook. There's another video you can watch about the Hershey-Chase experiment. So like I was saying earlier, when I was saying in the 30s they knew this, and in the 40s they knew this, in the 50s they knew this. Like I said, that's a good independent work topic. Who figured it out and how did they figure it out? And I won't say which, but that kind of scratches the surface for one of them. Here we go. The one you do need to know, speaking of who figured it out and how they figured it out, it's Watson and Crick. Your book calls it Watson and Crick. But as you're going to learn, there's more people involved than just Watson and Crick, and that's actually a big controversy in the scientific community because some proper people didn't get their, or some people didn't get their proper acknowledgement for their work in it. But anyway, let's jump into it. We're about to talk about how we figured out the structure of DNA, right? The double helix. That's what I'm about to teach you. Just like earlier, your book does it kind of like in a story fashion. Like, oh, first they did this, and then they did this, and this is how they figured it out. So as I'm teaching this, I want to remind you, this is not a history class. So I'm not going to ask you any questions like, what did they do first, and who did this, and who did that? It's not going to happen that way. But again, your book points out that it was Watson and Crick that determined that DNA is a double helix. What is a double helix? Well, we've said it throughout the semester, but the official definition is it's a spiral with uniform diameter made up of two polynucleotide strands. Who cares? I'm putting it next to that. I'm not going to ask you the definition of a double helix. The only reason you need to know what a double helix is because when I use the words, you need to know what I'm talking about. And another thing that's important to this whole idea of how DNA works is the fact that it is, and this is why I keep reiterating this point, that strand of DNA, that curvy thing that you always see, remember, that's actually two strands of DNA attached to each other with hydrogen bonds, which I'm about to teach you again. I taught it in chapter 3. I'm about to teach you again. But the fact that there's two of them is actually important. That is probably the most important part of the structure as far as the function is concerned, right? And that's what we're most concerned about is the function. And the whole middle part of this chapter is going to be how does it function? How does the DNA replicate? How do we go from DNA to RNA to proteins? So to understand that, you need to understand the structure. Again, as far as we're concerned for this conversation, the most important thing about the structure is the fact that that's two strands of DNA. So again, I'm not going to ask you any questions about what a double helix is, but I want you to focus in on that word double. So when you think of a double helix, like a double helix, who cares that it's a helix? The fact that it's spiraled means nothing to what I'm about to teach you. It is important in other contexts, but not for this. Anyway, into the story. Not that I'm going to ask you this, but Watson saw an X-ray image of DNA that had been produced by somebody named Rosalind Franklin, who I'm going to tell you a little bit more about later. And then after seeing that, he got together with Crick, and they compiled some other information that we don't have time to get into, and it's also not relevant for understanding this chapter. It's only relevant for historical purposes. But anyway, they used wire models, and they tried to construct the double helix that conformed to everything they knew about DNA, right? So they knew this from these people, and they saw the DNA image from Franklin, and they knew that from those people, and they compiled all this information and used wire models to figure it out. So really, they were standing on the shoulders of giants. It's not like they figured everything out, and that's usually how science goes. They they put all this other stuff together that other people had figured out, and then figured out something new from it. So again, as far as the exam is concerned, this is a very unimportant slide. But are there any questions about it? All right, there's Watson and Crick over there looking at their wire model. There's Franklin. Actually, it really is. If you get a chance, and maybe I can find a video to share with you, it is a very interesting story. They weren't exactly great students. They were pretty much slackers. They spent this is in England, they spent a lot of time in the pub, and they took a long time to finish their degrees, but they ended up being some of the most famous biologists in all of history, which I like. Anyway, the model of the DNA Looks like a ladder twisted into a spiral. We already know this. That's what we call the double helix. So if you were to think of this ladder as a strand of DNA, and you already know this, we're just looking at it from a different point of view to help solidify what you already know. These sides here would be the sugar phosphate backbone, right? Sugar, phosphate, right? Because they're all covalently bonded to each other. The rungs in between the two sides would represent the bases, right? The A's, the C's, the T's, and the G's. So that's all old news. There's nothing new here. The only new information is this bullet point right here, and there's no need to write it down either because we're about to jump into it. That bullet point in itself does not give you any important information. It's a segue into the important information, which is the base pairing rules dictate which bases go with which bases, right? So it's not random. It's not like, A's. Well, I'll go ahead and tell you, even though I'm going to tell you again here in a second. A's always go with T's, and C's always go with G's. So there's rules that dictate that, and that's what we're about to talk about. So again, even though there's a lot of information on this slide, first of all, from here up, it's just everything I've already told you. And then this third bullet point is just a segue into the important stuff. So there's nothing important on this slide for the exam. Any questions about this slide? Again, I'm just looking at it in a different way. So if we were to look at it as a model, right? The sides would be the sugar phosphate backbone for this strand, sugar phosphate backbone from that strand. These would be the nitrogenous bases, hydrogen bonded to each other. And of course, if you twist it, then it would look like what we think of as the DNA molecule. Not that any of that matters at this level. There's another picture of it. All three of those pictures show a DNA double helix. It's just they show them in a different way. There's a ribbon model, there's an atomic model, there's a computer model. They're all useful in their own ways depending on how you want to look at it. I'm certainly not going to ask you any questions. I'm not going to give you those three pictures and say, which one's the atomic model and which one's the ribbon model. Won't do that. That's just an example. If you download the PowerPoint, you can watch yet another little animation that talks about DNA double helix. In 1953, you know what? That's a good time to stop. When we get back, we'll finish up what Watson and Crick did. So the last word for attendance then will be, let's see, Nobel Prize. Nobel Prize. So if you're online, send a picture of a Nobel Prize winner. And I know that sucks. You're going to have to Google something. But send a picture of a Nobel Prize winner. And that's it. We'll see you on Friday.